This is the lecture for Chapter 20, Antimicrobial Drugs, Part 1. This chapter is all about controlling microbial growth, but it's controlling microbial growth in terms of controlling an infectious disease. An infectious disease is when a microbe has infected an individual, and infected means that it has invaded or colonized that person's body. Disease means that there is a change in the person's state of health. So an infectious disease means that an individual has been colonized by the microbe, and that colonization is causing damage to the person's body. When treating an infectious disease, we want to come up with a type of treatment that is selectively toxic. And what we mean by selectively toxic is that whatever that treatment is, it will not have an effect on the host cells, the eukaryotic cells, but it will have an effect on the microbe cells. So in the case of a bacterial infection, the bacteria are prokaryotic cells, and the human, if we're talking about a person, the human is composed of eukaryotic cells. So a good type of treatment would be one that would preferentially kill or inhibit the growth of the bacterial cells, the prokaryotic cells, but would have no effect on the eukaryotic cells. So in order to determine what types of medications can be selectively toxic, it's very important to understand the differences in structure between eukaryotic cells and prokaryotic cells. So often these different types of medication will target those structures that are found only on prokaryotic cells, and since the eukaryotic cells do not have those structures, the eukaryotic cells will be unharmed. This chapter covers different types of antimicrobials, but it spends most of the time on antibiotics. And antibiotics are specific against bacteria. The first antibiotic was discovered by Alexander Fleming in 1928. And that antibiotic was discovered because fungus had colonized a bacterial plate, and what he noticed is that fungus seemed to secrete something into the agar that prevented the growth of the bacteria. So he referred to that substance as an anti-against life biotic. And that first antibiotic was penicillin. So he discovered penicillin and the action of penicillin, but it took two other scientists, Florey and Chain. It took them about 10 years to actually take that molecule, that penicillin, and convert it into a usable form that could be used to treat infections. And penicillin didn't become widely used until about 1945. So really, we've only had antibiotics for about the last 75, 80 years. And since then, penicillin is not widely prescribed anymore because most bacteria are now resistant to it. This is a table that lists some common antibiotics and where they come from. So some of these are like bacitracin, also there's neomycin, and of course penicillin. And what you should notice is that all of these antibiotics come from microorganisms. So they are naturally produced by different types of microorganisms. Penicillin, of course, comes from a type of fungus, and then all these others come from bacteria, from gram-positive rods, and the actin-mycetes. Those are also a type of bacteria. So fungi and bacteria actually produce antibiotics. And why would the fungi and bacteria produce molecules that can kill other bacteria? It is because it will give those microbes a selective advantage. So the microbial world is incredibly competitive in terms of space and nutrients. And so if a species of microbe is able to secrete a substance to kill competing bacteria, that will give that species a selective advantage. So that is why there are so many different types of antibiotics that are produced by different types of microbes. Every type of bacteria has a particular spectrum of activity. And when we say spectrum of activity, we're referring to the types of bacteria that they target. In general, they can have a narrow spectrum or a broad spectrum of activity. Penicillin is an example of a narrow spectrum antibiotic, and it's considered a narrow spectrum of antibiotic because it only affects one type of bacteria, and those would be the gram-positive bacteria. Streptomycin is considered a broad spectrum, and it is a broad spectrum antibiotic because it can affect more than one group of bacteria. In this case, it can affect the mycobacteria and the gram-negative bacteria. Tetracycline is a very broad spectrum antibiotic because it can affect the gram-negative bacteria, the gram-positive bacteria, and also the chlamydias. So broad spectrum and narrow spectrum, it relates to the types of bacteria that are targeted by the antibiotic. A Kirby-Bauer assay is used to determine the effectiveness of an antibiotic, and this is a type of disc diffusion assay. To perform this assay, the first thing that would be done is a plate would be taken and a lawn of bacteria would be inoculated onto the plate. After the bacteria has been inoculated onto the plate, then a filter paper disc containing various antibiotics will be placed on the plate. During the 24 to 48 hour incubation, those antibiotics would diffuse through the agar, and if they are able to inhibit the growth of the bacteria, you will observe a zone of inhibition. And some of these antibiotics may be bactericidal, where they kill the bacteria. Others may be bacteriostatic, where they prevent the replication of the bacteria. Antibiotics are selectively toxic. That means that they will have a negative effect on prokaryotic cells and not on eukaryotic cells. And the reason they have these negative effects on the prokaryotic cells is because they target certain structures that are found only in prokaryotic cells. With all the different antibiotics that are produced by various fungi and bacteria, all these antibiotics can fall within 5 different modes of action. This figure will summarize the 5 different modes of action of antibiotics. The first mode of action is for antibiotics to have an effect on the cell wall. And having an effect on the cell wall will be bactericidal. Because remember, the purpose of the cell wall is to protect from osmotic lysis. When a bacterium is in the human body, the bacterium is hypertonic compared to the human tissues. So the direction of osmosis is into the cell, which means that water is moving into the cell, increasing osmotic pressure inside the cell. Normally the cell would rupture, but if it has an intact cell wall, then it is protected from osmotic lysis. Weakening the cell wall will leave that bacterium sensitive or vulnerable to osmotic lysis. Penicillins, cephalosporins, bacitracin, and vancomycin kill the bacteria in this way. They weaken the cell wall, so then the bacteria begin to rupture due to osmotic pressure. This is an example of a rod-shaped bacterium, which would be a bacillus, in a hypotonic environment before adding penicillin. And this shows the bacterial cell lysine because the penicillin has weakened the cell wall. Osmotic pressure has increased inside the cell and has resulted in lysis. And of course, this is killing the cell. It is bactericidal. The second mode of action is to inhibit protein synthesis. And basically, this means to stop the process of translation. So these different types of antibiotics, chloramphenicol, erythromycin, tetracyclines, and streptomycin, they target the ribosome because it is the ribosome that performs translation. As a review of ribosome function, remember that for a ribosome to translate, the first thing it needs to do is to read the codon on the mRNA. So it has to recognize the codon on the mRNA. Then the second thing it needs to do is match the appropriate tRNA to the codon. And remember, the tRNA has an anticodon on one end, which is complementary to the codon on the mRNA. On the other end of the tRNA is the appropriate amino acid that correlates with the codon. Once it has matched the appropriate tRNA to the codon, then it forms a peptide bond. And that peptide bond is linking together the amino acids. And ribosomes do this over and over. They would slide along, read the second codon, match the appropriate tRNA to that codon, and then the last thing they do is link the amino acids together by forming a peptide bond. Translation involves ribosomes performing 3 different individual processes to complete translation. So those 3 different processes are reading the codon, matching the tRNA, and forming peptide bonds. So because there are these 3 different stages of translation, that means there are 3 different ways antibiotics can interfere with translation. So an example is streptomycin. Streptomycin prevents the ribosome from reading the codon on the mRNA. Tetracyclines, on the other hand, prevent the ribosome from matching the tRNA to the codon. And chloramphenicol will prevent the formation of peptide bonds. So ribosomes are performing 3 different tasks during translation, so those are 3 different targets for antibiotics. And with all of these antibiotics, they are targeting the 70S ribosomes. So these antibiotics only inhibit the function of prokaryotic ribosomes. They will not inhibit the function of eukaryotic ribosomes. The third mode of action for antibiotics is the inhibition of nucleic acid replication. And this occurs in 2 places in the cell. The first example of nucleic acid replication is during DNA replication, where the one chromosome is replicated into 2 other chromosomes. So some antibiotics, like the quinolones, that can inhibit DNA from being replicated. The other example of inhibiting nucleic acid replication is in transcription. Remember, the purpose of transcription is taking the DNA and making a copy into RNA. So rifampin has that ability to inhibit that process. Quinolones are a type of antibiotic that can inhibit DNA replication. And the way it does this is that the quinolones bind to topoisomerases and DNA gyrases, which are important for unwinding and unzipping the DNA. So it prevents those proteins, those enzymes from functioning, which will prevent DNA replication. And a quinolone will be bacteriostatic. So bacteriostatic because it's only going to prevent the bacteria from replicating their DNA. And bacteria, or all cells, only replicate their DNA right before they divide. So the only thing the quinolone is doing is preventing bacterial replication. Rifampin, on the other hand, inhibits transcription. And what rifampin does is it binds to the RNA polymerase and it prevents the RNA polymerase from transcribing the genes in the DNA. So if transcription is inhibited, then there won't be anything to translate and there won't be any protein production. If there is no protein production, then that is bactericidal, because bacteria need to be able to produce enzymes to facilitate their metabolism. So if there is no transcription, there is nothing to translate, that will result in the death of the cell. The fourth mode of action is to damage the plasma membrane. And an antibiotic that does this is polymyxin B. And the plasma membrane, remember, it's a vital part of cellular structure. It serves as a selectively permeable barrier to separate the inside of the cell from the environment, yet it allows nutrients to cross into the cell and waste products to cross out. This is a figure that shows the action of polymyxin B, and for this it shows a gram-negative bacterial cell wall. You know this is a gram-negative bacterial cell wall because there's an outer membrane and one layer of peptidoglycan. So what will happen is that the polymyxin B will bind to and infiltrate into the phospholipid bilayer, and it will disrupt the phospholipid bilayer, disrupting the membrane and causing the death of the cell, because all cells need an intact cell membrane to function. The last mode of action is to inhibit synthesis of different important molecules. And to inhibit the synthesis of important molecules, basically that means inhibiting enzyme activity. So antibiotics like the sulfonamides, they act as inhibitors for different enzymes. The sulfonamides act as a competitive inhibitor on an important enzyme that produces folic acid. So normal folic acid formation, it involves this enzyme binding PABA and producing folic acid. The sulfonamides have a similar structure to the PABA. So if the sulfonamide is present, the sulfonamide will bind to the active site in the enzyme, and when it binds there, then that inhibits the PABA from binding, so there will be no synthesis of folic acid. And folic acid is an important metabolite that is required by the bacteria. So if the bacteria are unable to form folic acid, then they will die. So sulfonamides are bactericidal. So as a summary of the different modes of actions for antibiotics, there are 5 different targets that the antibiotics can have. They can target the cell wall, they can target the ribosomes, which performs protein synthesis, they can target the enzymes that perform nucleic acid replication, they can target the plasma membrane, or they can target enzymes and prevent them from functioning. All of these targets are things specifically found in the prokaryotic cells that are not found in eukaryotic cells. So cell wall, eukaryotic cells, animal cells, human cells do not have a cell wall. Protein synthesis is the ribosome, and remember that ribosome measures 70S, so those antibiotics will only bind to a 70S ribosome. For the nucleic acid replication, those proteins, those enzymes that are involved are different than the eukaryotic enzymes. So those antibiotics only target prokaryotic enzymes that are involved in nucleic acid replication. Injury to the plasma membrane, that polymyxin B is recognizing lipids that are specific to the prokaryotic membranes. And the last example, the enzymes that are involved in various biochemical reactions, those enzymes are specific for the prokaryotic cells. So all of these antibiotics will specifically target the prokaryotic cells and have very little effect on eukaryotic cells. So these antibiotics are selectively toxic. And all of these antibiotics are bactericidal. They will result in the death of the bacterial cell, except for quinolones. Quinolones, they prevent DNA replication, which is only required for a bacterial cell to replicate. So if the DNA cannot be replicated into 2 daughter molecules, then the bacterial cell cannot divide. But otherwise, the bacterial cell can still function. So the quinolones, or preventing DNA replication, is bacteriostatic. It will prevent the bacteria from replicating.